All right, thank you, everybody. I'm Bryan Frazier. I'm from the South Carolina Department of Natural Resources, and this is actually a part of a, what did I just do? Part of a master's thesis by Bobby Edmond. So I'm just going to be presenting his work. Just to give you background on tiger sharks, their circumglobal distribution, primarily found in tropical and temperate waters. And in the North Atlantic, we know they have this nearshore to open ocean movement. More recently, folks have described and recommended that this green area be added into the tiger shark distribution based on fishery observers data. And based on recreational recapture data from Kohler et al., we even see transatlantic movements by these tiger sharks. There has been telemetry work done on tiger sharks by Hammerschlag in the Bahamas as well as South Florida, and by Lee, which I think Matt brought up out in the Bermuda area. But where I am here in South Carolina, we don't really see any detection data, and certainly not from the lead tiger shark, from the lead sharks either. So I really got interested when we moved down. We have some fishery independent sampling in South Carolina, and in 2004 we moved down and started longlining in these two sounds, and we started catching a lot of tiger sharks down there, and in this kind of estuarine interface area. At the same time, a charter captain who was working down there was catching a lot of tigers, and he actually was the one who approached OCEARCH. And they kind of were working on some other stuff at the time, and actually put him in touch with me. And OCEARCH facilitated this research starting by hooking me up with the captain, but also providing us with spot tags to begin this study. And our two study sites are here in Port Royal Sound in Hilton Head, and then St. Helena Sound up here. And as I said, these are high salinity estuaries. One is very deep, one is very shallow. The objectives of our study were to record the horizontal movement patterns of these tiger sharks. And we were primarily trying to do this through spot tags as well as acoustic telemetry. And within that, we wanted to see if there was any site fidelity to these sounds, look at seasonal movements, dial movements, how they were moving in response to lunar and tidal cycles, and then see how they were really using these estuaries. While we were doing this, we wanted to accomplish as much as possible, so we incorporated some stable isotope analysis into our study, and it also allowed several other studies, including the non-lethal reproduction tiger shark work that UNF and Chelsea Shields are doing that Jim mentioned, some genetics work, and your next talk you're actually going to hear about some of the antimicrobial bacteria work. So, similar to Matt, we are catching these using drum lines as well as rod and reels. We get them to the boat, we secure them, we implant an acoustic transmitter, we draw blood at that time, we take any other samples that we're going to take, fin clips, muscle biopsies, and if we had one, we applied a spot tag. So to date, we have a total of 47 sharks in our studies. These are larger juveniles to adults. Slightly a 2-to-1 female to male ratio. 45 of these have been implanted with an acoustic transmitter, and 17 were double tagged with a spot. To look at our size distribution, this is a little bit unique in the rest of the Atlantic studies. In the Bahamas, all of their sharks are females. And within the Bermuda tag sharks, they were mostly males. So we actually have a nice ratio of both immature and mature sharks, as well as male and females. We have more mature females by far, and then we do have some representative mature males, but also a few more immature males than mature. One of the cool things about our study is we were able to get a lot of recaptures, both of when we started working and initially tagging these animals, but also recapturing our tagged animals within the study. So of our initial encounters with animals, 10 had actually been previously tagged, and 9 were tagged in Port Royal Sound. One was actually tagged in the Gulf of Mexico by Mote Marine Lab, and we'll talk about that one a little bit later. And we were actually able to recapture one of our spot tagged individuals, and also 4 of our acoustic recaptures. And the great things about those recaptures is we were able to serially draw both blood and muscle, which for those non-lethal repro studies and stable isotope studies will really give us some great information on interannual variation. So looking at our spot tag data, unfortunately, with our tiger sharks, we don't get great tracks. Our maximum track is 197 days, and our average track is less than 2 months with these tigers. But we have gotten some really interesting data. Primarily what we're seeing is coastal and shelf water movement. Uh, and we're seeing a lot of these individuals, some of them going up the coast, but most of them sticking around here in the southeast. We see both directional and restricted movements among individuals. So this would be restricted movement after tagging, and then it migrates up here up to North Carolina and then settles in this area. And as I said, you know, a lot of these individuals are going up the shelf, and when they are going up the shelf, they seem to be following right along this continental shelf to migrate. And of ours, only 3 migrated up the southeast coast, and the rest of these actually stayed all summer in the southeast, at least for our spring and summer animals. For our autumn-winter animals, we generally see a southward movement as water temperatures cool. They're moving a little further off the shelf first, and then if water temperatures really cool, we see them move down towards the Bahamas or further south off of Florida. And analyzing these data, we really see that temperatures are driving these movements, and we see that they prefer a sea surface temperature above 19 degrees Celsius. So we wanted to look at these data versus the nearest tagged tiger sharks, which are hammerschlags. And he then generated these kernel density estimates. And basically what you're seeing is the red indicates more preference for an area based on the spot tag tracks. Matt talked about a little bit about how these data are analyzed. And we are seeing some differences from theirs. We are seeing some overlaps in these areas off of North Carolina here, as well up off the northeast. But his tiger sharks generally did not come within these South Carolina coastal or nearshore waters, whereas we saw ours had a very strong affinity for these waters here. We are a little bit limited by the duration of these tracks. However, for this analysis, we only included data after at least 23 days after tagging to remove a little bit of those biases of that tagging location. Moving into some of our acoustic data, we primarily got our detections from the southeast U.S. and the Bahamas. Minimal detections on other arrays. We did have some detections from up in Virginia, some detections down Fort Pierce, Florida, but primarily in these few arrays, and they are porous arrays. You know, we shouldn't say that these arrays, you know, we can say with any certainty that we detect an animal that's there. I mean, a lot of these receivers are a mile apart, and we're talking about a detection range sometimes because of turbidity and things of less than 200 meters. Most often probably between 100 and 200 meters. So looking at our acoustic detections, this is a lot, so I'm actually going to pull out a few examples and kind of break it down. But what you can see, so the X's indicate when we tag the animals. What you can see is, we've been able to add to our study every year. And here, actually in the fall, we added this St. Helena sound array, which unfortunately we didn't have all our arrays in place, and we've been kind of building this project as we could, but we really see that color show up a little bit later. So when we analyze these data, 2 things we were interested in is diel and lunar patterns. And we do see across almost all arrays that we do have increases in the amount of detections during the day. And this is our Charleston array, Port Royal Sound array, as well as the Bahamas, especially in the Bahamas. It's very striking when they show up. We also see an increase in detections during new and full moons. And this is a little bit surprising because this is when we have greater tides in South Carolina. The water's a lot more turbid, a lot more water movements. You would think that detection range would go down, but we're actually seeing an increase on both these new and full moons. And this is likely indicates they're coming in for some sort of opportunistic feeding, perhaps spawning of some teleost fish or something like that. So looking at individual movement patterns, these are 2 individuals that we would consider residents to areas showing a high degree of phylopatry. This one here, this is the most interesting one for me because this individual was tagged as a 1-year-old tiger shark off of Sarasota Pass by Mote Marine Lab. 7 years later, we recaptured it in Port Royal Sound and we stuck this transmitter in it. And this thing has been in Port Royal Sound, migrating out but coming back every year since we tagged it, and has only been detected in one other array in all of that time. And it should be a mature female now. So very interesting, that it was, initially tagged in the Gulf of Mexico and has fidelity to another area now. And then this red color here actually represents Tiger Beach in the Bahamas, and this individual alone accounted for more than half of our detections across the entire study. What's interesting is we tagged it, it went to Tiger Beach, came back and was detected back in the Sound, went down there again, and then skipped a year, didn't come back, and spent an entire year there. Most of their tigers actually leave during the summer, and then actually came back again this year. So interesting there. And then we see some movements that we'd like to call visitors, and these are likely the individuals that we see migrating up the coast to where we get some detections. They show up for a day, get a couple detections. We don't see them for another year, get a couple detections, don't see them for another, a couple years. And one of these we actually know was one of our spot tag tigers that moved up the coast, and that was Septima. So this kind of confirms that she has that migratory behavior. And then we see a few that we know or we believe were either lost to us either through fisheries mortality or perhaps just shift in habitat preference. Two of these are spot tag ones. Our most famous spot tagged tiger was Chessie. And things were going along great, and she was pinging great and actually swam all the way up off of New York. And then just her spot tag completely ceased transmitting. And we haven't gotten a detection from her since. And we do know, that we are having a lot of encounters with our individuals. This was actually this summer, and I'm not sure if you can see, but this individual was killed in a shark tournament right off of St. Helena Sound. And if you look closely at the dorsal fin, there's a chunk missing that indicated it likely had a spot tag at some point. And based on our acoustic detections, we believe this was our spot tag tiger, Jax. We had recent detections of it and haven't detected it since. So, coming back to this full map, what we see is, this we've had the array in place in Port Royal Sound since the study started, but as I said, we just added last year that St. Helena Sound array, and in the first year alone, we had over 50% of our sharks detected in that sound, and if you combine that with the Port Royal Sound, over 80% have been detected just in this May-June period last year. So they're definitely returning to this habitat every year. We think it's likely opportunistic feeding. These areas are both very important in terms of shark pupping, which occurs in May and June. They're primary nursery areas for a variety of small and large coastal species. And at the same time, we know that tiger sharks feed on horseshoe crabs, and we see a lot of horseshoe crabs, especially around those full moons coming up on the beaches in these sounds to lay their eggs. So they could be taking advantage of that food resources. And we also, which you can't really see in amidst all this noise, but we do get another pulse of detections in October, November in these sounds, and we believe that is them coming back, still taking advantage of food resources, but we know that mating is occurring. We do see a lot of these tiger sharks with very fresh mating wounds, and we're looking forward to being able to analyze and pair this up with some of Chelsea's data to see if we can work out some of the reproductive cycle with some of these individuals, especially when we get some. We've had a couple that we've gotten mating wounds on, been able to draw blood, and actually recaptured them a year later. So it's going to be really interesting to look at those data. So kind of wrapping this up, we see two primary behaviors in our tiger sharks. We see these animals that come up and they spend the whole summer in the southeast U.S., primarily off of South Carolina and Georgia. And then we see these animals that are migrating up the coast. But all of them seem to be, and this is based on these limited spot tracks, but all of them seem to be staying very close to and on the continental shelf. We do see phylopatry in limited individuals, but we also see this diversity of behavior. And this is seen across tiger sharks, where some individuals are highly fidelic to an area and some individuals just seem to wander. We do see mating, and we believe it's likely opportunistic feeding that's bringing them to these areas. We do see a relatively narrow temperature range. Something I didn't really pull up, but we actually see a marked decrease in detections in these sounds in July and August. And we believe this is actually them avoiding those hotter water temperatures. We really see that they prefer these 19 to 26 degree temperature ranges. And once that water gets up to 30, 31 degrees, we're not really seeing any tiger sharks in these waters. And our work really kind of highlights this importance of tagging sharks at multiple locations. Right? If you were to take any one of these tiger shark papers that has studied tiger sharks in the Atlantic, if you were to take them in isolation, you're going to draw some very different conclusions about what areas are important to tiger sharks. And so, the more data we have, the more areas we can tag these individuals in. We can take a much broader look at tiger sharks and certainly bring in all these different data sources and perhaps come up with something for the entire Atlantic, bring people together and really come up with some good conclusions. Our tigers, they're definitely going to have a much higher encounter rate with fishermen than these other fish. They're strongly coastal. A lot of these guys have 2 or 3 hooks in their jaws when we get them. We've seen them trailing commercial gear. So we know, that they're likely to have a higher mortality rate than some of these open ocean tigers. And one of the unfortunate things is spot tags aren't great for these sharks. We think they're actually, because of the shallow water that they're constantly in, they're able to go down, they're able to dive and actually rub these tags against the sand. The one recaptured spot tag we had looked like somebody had taken 100 grit sandpaper to it. So we think they're likely damaging them. And, perhaps we need to experiment with some toad design, something like that, that, they can't necessarily damage. So with that, I'd like to kind of, acknowledge everybody, especially OCEARCH, who facilitated this research. We've been able to kind of piecemeal this together with private donations, and it's something I hope we can keep going for the long term. We're certainly going to write this up, hopefully this spring. But we've got 7 to 10-year pingers on these tigers, so we're going to be able to monitor their movements over the long term. A lot of people have contributed acoustic data as well. I didn't talk about stable isotopes, but we're working through that, and hopefully we'll be publishing that as well. So with that, any questions?